So, welcome to today's session of Plant Physiology and Metabolism course. Today we will study very fascinating aspect of that is fermentation. So, you see that we have gone through into detail about glycolytic pathway which generally could takes place in presence of oxygen. So, today's discussion we will discuss about fermentation and we have already gone through in detail about the glycolytic pathway and you see that in glycolytic pathway the end product is pyruvate and then what is the fate of pyruvate under aerobic conditions. Now our focus now should shift what happens to this pyruvate under anaerobic conditions, or you can say what are the different fates of pyruvate under aerobic condition, and this is known as we will discuss under the fermentation. Especially what is the fate of pyruvate under anaerobic condition known as lactic acid fermentation, and what is the fate of pyruvate under alcoholic fermentation. So, first we will explore the discovery of fermentation and its application in industries and what is its commercial significance. So, all these aspects we will discuss into detail. So, now we see that why fermentation or what is fermentation. You see that in condition when plants are generally under submerged condition, then plant have depleted level of oxygen and that plant face generally anaerobic environments, and at this in this situation what happens generally respiration of root is generally inhibited due to lack of oxygen which is generally required for mitochondrial electron transport chain we have discussed in our previous lecture. So, now what happens without oxygen generally which serves this oxygen serves as a terminal electron acceptor and then in this condition NAD cannot be oxidized then disrupt the regeneration of NAD. So, this absence of NAD basically hinders the oxidation of glycerylate 3-phosphate which is a very key step in the process of glycolytic pathway. So, generally it halts energy yielding reactions. Now, what could be the remedy? How glycolysis can continue in the absence of oxygen? So, in such a situation or you can say in absence of oxygen generally glycolysis adopt alternate pathway which is needed for regeneration of NAD. So, under anaerobic conditions what happens basically pyruvate which is the end product of glycolysis is metabolized through lactic acid or ethanol fermentation. So, basically this process allows the oxidation of NADH to regenerate NAD. In plants, the primary fermentative pathway involves the breakdown of carbohydrates into ethanol and carbon dioxide, and this adaptation basically enables plants to survive and generate energy under anaerobic environments, for example, as waterlogged soils or during the process of seed germination. Now, what is the fate of pyruvate? Pyruvate, you see that this is the end product of glycolysis, is metabolized differently under aerobic condition and anaerobic conditions. So, we have gone into details what happens to the pyruvate under aerobic conditions. Basically, under aerobic condition, this pyruvate is oxidized to acetyl coenzyme A in mitochondria, which then enters the citric acid cycle and the NADH formed in the glycolysis is then oxidized to NAD through NADH shuttle system, and NADH is basically not transported across the inner mitochondrial membrane directly because of its transporter are generally absent. Under anaerobic conditions, pyruvate is metabolized through the process of fermentation. Now, what happens under anaerobic conditions? You see that anaerobic conditions has been observed in active skeletal muscles in human being, submerged plant tissue, solid tumors or lactic acid bacteria, where you see that the availability of oxygen becomes a limiting factor. So, what happens when oxygen is present in low concentration? At this situation, the NADH generated by glycolysis encounters a sort of challenge, as it cannot effectively reoxidize by oxygen. So, this poses basically a significant issue, as the regeneration of NAD is very crucial for sustaining the continuous progression of glycolysis, and which is also essential for energy production. So, to circumvent or to cope the absence of oxygen, cells generally have evolved very specific mechanisms to regenerate NAD during anaerobic glycolysis. So, one common strategy generally involves the transfer of electrons from NADH to other molecules, resulting in the formation of reduced end product. So, depending on the type of organisms, these reduced end product can manifest as lactate or ethanol, and this electron transfer process generally ensures the continued regeneration of NAD, thereby allowing the glycolysis to persist even in absence of oxygen. So, this is very crucial to note at the moment. Now, you see the ability of cells to adapt to both aerobic and anaerobic conditions is very crucial for their survival and efficient energy production, and then this also is very crucial for evolutionary adaptation of organisms to diverse environmental conditions. Initially, during the lactic acid fermentation, what happens? Generally, oxygen deficiency induces the synthesis of lactic acid dehydrogenase, and then you see that in the slide, pyruvate is converted to L-lactate by the enzyme catalyzed by lactate dehydrogenase, and you see that lactic acid formation generally lowers the cytosolic pH, resulting in inactivation of lactic acid dehydrogenase and upregulation of alcohol dehydrogenase. So, oxidation of NADH to NAD is generally coupled with the reduction of pyruvate to either lactate or ethanol, and there is no net change in the oxidation state of carbon in glucose when converted to lactate. Most of the energy is still retained in the molecule of lactate. During fermentation, there is a net production of 2 ATP for every molecule of glucose being converted to either lactate or ethanol. So, this is a very interesting point to note. Now, alcoholic fermentation is a metabolic process that converts pyruvate, a product of glycolysis, into ethanol and carbon dioxide in the absence of oxygen. So, this process is generally commonly used in the production of alcoholic beverages and generally carried out by adopted by certain microorganisms, including very brewer's yeast, very interesting example, Saccharomyces cerevisiae. Now, we will discuss basically the basic steps involved in alcoholic fermentation. First, decarboxylation of pyruvate takes place. You see that you are already aware that pyruvate is a 3-carbon compound, which is end product of glycolytic pathway. Now this pyruvate is decarboxylated in an irreversible reaction by the enzyme pyruvate decarboxylase. So this reaction results in the removal of, you see, carbon dioxide molecule from the pyruvate, producing acetaldehyde. Now what is the role of pyruvate decarboxylase in this reaction? You see that pyruvate decarboxylase basically catalyzes the decarboxylation of pyruvate and requires the presence of magnesium ion. Here you see it requires for its proper functioning the presence of magnesium ion and another cofactor called thiamine pyrophosphate, that is TPP. Now once this acetaldehyde is formed, it is further reduced to ethanol. So alcohol dehydrogenase is a very peculiar enzyme responsible for reduction of acetaldehyde to ethanol, and this conversion of acetaldehyde to ethanol basically involves the transfer of electrons from NADH to acetaldehyde, resulting in the production of ethanol and oxidation of NADH to NAD. Now what is the end product of this ethanol fermentation? So the final product of ethanol fermentation are basically ethanol and carbon dioxide, and you see that after going through the alcoholic fermentation, generally it involves the conversion of pyruvate to ethanol and carbon dioxide through a series of enzymatic reaction. And this process basically is utilized in the production of alcoholic beverages, and generally similar enzymatic steps generally occur in the metabolism of ethanol in organisms including also human being. Now I will take you back little bit, a flashback in the history of this process of fermentation. Basically C. Wiesmann in the year 1910 was the first president of Israel. Generally he was the person who made a groundbreaking discovery of this process of fermentation. He found that certain bacterium like Clostridium Aztobacterium, this bacterium has the capability to ferment starch and then producing butanol and acetone. So this was a very significant and discovery which opened the door to the field of industrial fermentation. Now question arises, what is industrial fermentation? So with this discovery marked the, you can say, inception of industrial fermentation, and actually what happens in the industrial fermentation, readily available carbohydrate rich materials like cornstarch or molasses are supplied to a specific pure culture of microorganisms, and the microorganisms then ferment the material, transforming it into a more commercially valuable product. And generally this microbial fermentation is integral to the production of ethanol for gasohol and various acids such as formic acid, propionic acid, butyric acid and succinic acid. It basically also contributes to the production of glycerol and methanol, isopropanol, butanol and butanediol. So these fermentations generally takes place in a sizable closed vessels or vats, and where temperatures are carefully controlled, and basically limited access of air which promotes the desired growth of microorganisms and also helpful in preventing microbial contamination, other microbial contamination. So basically fermentation is a sort of a very old practice mastered by our ancestors 100 years back, and which plays a very crucial role in both food production and preservation. So you see that by harnessing the potential of microorganisms present in the raw foods, carbohydrates basically undergo fermentation, leading to creation of distinct textures and flavors. You see, you might be using yogurt, which is a well-known product dating back to old times, is a very interesting example of this fermentation process. Basically, the bacterium Lactobacillus bulgarius ferments milk carbohydrates, generating lactic acid and lowers the pH, causing protein to precipitate. And this results to in the thick consistency and tangy flavor characteristics of unsweetened yogurt. Now, another example of fermentation, there is a fermentation carried out by one bacterium P. frudenrechi in the milk, which results in the production of propionic acid and carbon dioxide. So, basically propionic acid causes milk proteins to precipitate, while carbon dioxide creates characteristics holes in Swiss cheese. Various fermented foods including pickles, sauces, soya sauce, kimchi and dahi are produced through fermentation process all over the world. Basically, the pH drops during fermentation, which helps in food preservation by inhibiting the growth of microorganisms which cannot thrive in the prevailing low pH environments. Now, another example of fermentation in agriculture, plant byproducts such as corn stalks are basically preserved by use of animal feed and then use as animal feed by packing them into large containers, and these containers known as a silo with limited access to air. Basically, microbial fermentation basically helps to produce acids that lower the pH, and then the stored food that results from this fermentation process can be stored and kept as animal feed for long period without any sort of spoilage. So, thank you all for patiently listening this process of fermentation. I discuss in detail the different types of fermentation take place and how this fermentation could be exploited for industrial purposes. Thank you all. Thank you very much.